Good morning, students. So today in this lecture we will study about pedigree analysis. So what is pedigree analysis? What are pedigree charts? These are the diagrams which show the phenotypes or genotypes for a particular organism and its ancestors. So while commonly it is used in human families to track the genetic diseases or any traits, they can be used by any species and any for finding out the inheritance of any trait among different species. So, pedigree are often used to determine the mode of inheritance of any genetic disease, whether it is dominant or recessive among the individuals of a family, and how it is being passed from the parents to their children and their children to further their children. So, how it is being passed in a family. Now, geneticists use standard set of symbols to represent individual sex, family relationships, and phenotype. So these diagrams are used to determine the mode of inheritance of any disease or trait, to predict the probability of appearance of that trait or disease among the offspring of that family, right? So we just use these diagrams to find out how the trait or any disease is being passed in a family, how many individuals are affected by the trait, and what is the probability of that trait being passed on to the children. So pedigree analysis is an important tool for both basic research and genetic counseling. Now, why do we need to carry out pedigree analysis? For the organisms which have large number of offsprings and controlled mating, we simply can use Punnett square or chi-square test to predict the inheritance of the trait. But for the humans, where there are small families are there, even the large human families which earlier used to have 20 or few children were there, and no controlled mating can be carried out between the humans. So the geneticists have uh, devised this technique of pedigree analysis to find out the inheritance of genes or traits or disease uh, among the individuals, right? So we have studied, like in case of neurospora and drosophila, uh, the uh, the genetic study has been carried out in them. They have been used as model organisms for studying the inheritance of any trait or disease. Since we have already studied that drosophila, with humans, in between human and drosophila, number of genes, disease causing genes are found in drosophila, which are found in humans also. So although we have been using certain model organisms for carrying out the study about the genes, but with humans we can use this pedigree analysis to find out the inheritance of any trait or disease, right? So pedigree analysis is done by geneticists for individuals if there is a family history of one specific disease, or they show symptoms of any genetic disorder, or if the person is concerned about passing on a genetic problem to their children. So in that case, that person can go to a geneticist for carrying out the pedigree analysis. Now there are certain signs and symbols which are used in pedigree analysis. A square is used for a male, a circle is used to denote the female, and a marriage between a male and a female is denoted by a horizontal bar between a square and a circle. Then the children born from that marriage is denoted by a line drawn perpendicular to the marriage bar, and then the children born are further denoted by a square for a boy and a circle for a girl. Then dizygotic twins, that is non-identical twins, are denoted by these symbols, and monozygotic twins, that is identical twins, are denoted by this type of symbols. Then if the sex of the children is unspecified, then we use diamond shape uh, symbol. Then the number of children is denoted by the numbers written in between the circle and the squares. Then affected individuals are denoted by the shaded or darker circle and the squares. Then we denote the heterozygotes for any autosomal recessive disease by a half shading the square and the circle, right? Coloring the half of the symbol black, we can denote the heterozygotes. Then carrier of any sex-linked recessive disease can be denoted by a black dot in the middle of this symbol. Then death can be denoted by putting a horizontal line along the square or a circle. Then abortion or stillbirth is denoted by this symbol. Then propositus is denoted by a arrow, putting a arrow pointing at a particular affected individual. This arrow is actually added at a particular affected individual to indicate that this is the person who brought the trait to the geneticist attention. That is, we just consider this individual who brought mm, the trait in the attention of the geneticist, and from this propositus we draw the this pedigree chart. Taking this at the center, from this we just draw the pedigree chart of his ancestors and even for his children. Then we denote the first generation by generations by the Roman numerals. That is first, that is for the first generation, Roman number 2 for the second generation we use, and the children of same generation is denoted by the number 1, 2 and 3, like that. Then we denote the consanguineous marriage, that is marriage between the two cousins or relatives, by two double horizontal bars between a square and a circle, right? Now let's take an example of this pedigree chart. This pedigree diagram or pedigree chart is for cystic fibrosis trait. Then in this trait, we can, from this chart we can see this is a square which denote for a male and circle denote for a female. We are using numeral Roman numeral I for the first generation, Roman numeral 2 for second generation, and 3 for the third generation. And the marriage between the male and female of first generation is denoted by this horizontal bar, and by this vertical bar we are drawing the children born from this marriage. Again, circle for a daughter and a square for a son, circle for a daughter. Now this daughter is married to a man, which is again denoted by this horizontal bar, and this daughter is again married to a man, which is denoted by this horizontal bar. And this is how we prepare the pedigree. This is the children born from this individuals, and these are the children born from this individuals. Now since this black completely shaded or blackened square and this blackened circle denotes the affected individuals, right? So from this pedigree chart we can just find out the genotype of the individuals of different generations. For example, if this completely darkened square indicates that this person is affected, and this shows that both the parents, that is I1 and I2, are heterozygous for the trait, because if they were homozygous then trait would have been found in both of them. And since both of them are heterozygous, then only their one of the children is affected, others are non-affected. This shows that the trait has been passed down to this children from the parents, and not to this children. Again, in the third generation, again we find affected individual. This again indicates that second generation, fourth individual, I mean the third children born from this marriage, is also heterozygous, that is carrier in nature, because this girl is married to a man. Both of them must be heterozygous, then only the trait must have been passed to their children, and this one is affected. This shows that the trait is in homozygous condition to this children, and both the individuals, that is second generation, third children born to this parents and this man, both are carrier for the trait, heterozygous in nature for the trait or disease, and that is why the third generation one of the individual is affected. So this is how we predict the genotype of all the individuals belonging the uh, uh, to the pedigree analysis chart, pedigree diagram, right? And this since these two in this individual and marriage between these two individual, all the children are unaffected, so there must be, like, they must be healthy individuals, they must not be carrying the trait. Now the category of inheritance. Autosomal inheritance means it is inherited, the trait is inherited on chromosome 1 to 22, while sex linked means the trait is inherited on either X or Y chromosome. The examples of autosomal recessive trait are phenyl ketonuria, and then Tay-Sachs disease and albinism. Then autosomal dominant example is Huntington's disease. Then X-linked recessive means the trait is in recessive condition, it is attached, this trait is present on the X chromosome. It means this allele is found only on X chromosome and can be in males or females. Example is color blindness and hemophilia. Then X-linked dominant, it means this allele is found on X chromosome and can be in males or females. Example is hypophosphatemia. Then Y-linked means the allele is found on Y chromosome and can only be in males. Now let's see an example of autosomal recessive pedigree. So this type of trait is rare in the pedigree. This trait often skips generation because since this trait is with the recessive allele of the gene, for that allele to be expressed, since it's a recessive allele, it has to be present in a homozygous condition, then only that trait will be expressed. If it is present in heterozygous condition, the trait could skip generation and will remain hidden in the carriers. So heterozygous individuals would be carrier for the trait. Now this trait affects males and females equally, and possible disease include cystic fibrosis, sickle cell anemia, and phenylketonuria, Tay-Sachs disease. Now rare recessive traits are more likely to appear in pedigree when spouses are related to each other, that is consanguineous marriage, for if they are first cousins. This increased incidence occurs because relatives share allele by virtue of their common ancestry. If they have common ancestry, the alleles of the genes are same among the individuals. Siblings share 1/2 of their allele, half siblings share 1/4 of their alleles, first cousins 1/8 of the allele. Thus marriage occurring between such relatives, then they have a greater chance of producing a child who is homozygous for a particular recessive allele than do the unrelated parents, and thus the trait will be passed on to the child and child would be affected with that trait. Now autosomal dominant pedigree. This trait is common in pedigree. The trait is found in every generation since it is dominant allele of the gene. Then dominant will express whether it is in heterozygous condition or homozygous condition. Therefore this trait is very common in pedigree. It is found in every generation. Affected individuals also transmit the trait to about half of their children regardless of the sex because it is present on autosomal chromosome. There are few autosomal dominant human diseases, but some rare traits have this inheritance pattern. For example, achondroplasia, that is skeletal disorder causing dwarfism, right? Now dominant traits that are associated with reduced viability or fertility never become frequent in population, right? means dominant traits which are associated with reduced viability or fertility, then that they never become frequent. So most of the people who show such traits are heterozygous for the dominant allele, and if their spouses do not have the trait, then half of their children should inherit the condition. Now, this is again an example of autosomal dominant trait, Huntington chorea disease. Half of the people in Venezuela village Barranquitas are affected with this trait. Since it is an autosomal dominant, it will express whether it is in homozygous condition or heterozygous condition. Therefore, it is found in large amount of people. So, a large scale pedigree analysis was conducted, including 10,000 people. So, it was found among large amount of people. So, this is all about today's lecture about pedigree analysis, and I will continue this topic in my next lecture. Thank you.